Welcome to Be the Spark, a podcast where we discuss immunology and the science driving that work. I'm Markus Mihalic, R&D Manager at Thermo Fisher Scientific, and today I have the pleasure of introducing Malia Edwards, Associate Professor of Ophthalmology in Baltimore, Maryland. Hi Malia, welcome to the show.
Hi, Marcus. Thanks very much. Very nice to be talking with you today.
So you are here at the Johns Hopkins Wilmer Eye Institute now. So anything you can think of what is unique about the environment and the location?
Yeah, so at Hopkins, people are studying everything. So we have the advantage of people studying multiple diseases, but we also have at the Eye Institute, we are all studying the eye with world-renowned experts in vision research that share the same passion and drive to really understand retinal diseases and diseases of the cornea, all parts of the eye, and eventually try to cure some of the blindness that happens in the world. So
Yeah, so a lot of collaborations, I guess, a lot of
yes,
great
exactly. Yeah.
networks. So let's jump to your research. So
Mhm.
what are the goals of your research projects?
So our focus are the glial cells. So we focus on two different types, the macroglial cells more than the microglia. And we look at these macroglia and their roles, they've often been thought of as more of a support cells. But in your central nervous system and also in the retina. And what's we've seen happening, things kind of happen in other neurodegenerative diseases, that these glial cells have a more of a role, like they're what it was often thought of as, oh, it's just a secondary effect, is actually important. And what we found is that there's some really drastic changes, what we call remodeling, where the cells actually change their structure and go into parts of the retina, move throughout the retina to places where they don't normally go, and change their location, change their protein distribution in response to retinal injury. And so I'm really interested as far as the glia, they're, what's happening to them and how are they really, when they remodel, do they maintain their normal function or are they losing that? And the glial cells are very important for neuroinflammation, which is where we get into more the immunology side. They can send signals to bring in a lot of other cells like the microglia and macrophages as well. And then the other side of our lab is studying mast cells, which is another understudied cells in the retina, also very important for the immune cells. So I think looking at these understudied cells, we can hopefully get a better understanding of the disease pathology and maybe unravel some novel targets for treatment.
It's highly interesting, and thanks for the detailed explanation and the different aspects of your research studies. In general, what techniques and applications do you and your lab use most frequently?
Yep, so we do everything. We're very fortunate that we get a lot of human donor eyes, and with that, we do a lot of immunohistochemistry. We do some RNA analysis with that and some protein, but mostly that's immunohistochemistry. We have a couple animal models that we work on, and there we're able to manipulate a little bit more. So there we can do RNA. We've done some single cell sequencing. We've used the Procardiplex assay there to look at the different cytokine levels and to look at how these cytokines might be changed in different stages of the disease. So that's going to be really informative to us as we try to understand where the processes that are happening and maybe the secondary effects. And we use we have cell culture models, both human tissue, iPSC derived, RPE cells. And that's again where the Procardiplex assay really comes into play and lets us really see, the ability to look at, especially when we have human samples that are so precious. If we can look at, up to 65 analytes in one sample with 25 microliters, as opposed to an ELISA where we use 100 microliters per sample, that's huge. And even when you're talking cell supernatants where you have a lot more, that gives us a much more robust answer and gives us, a lot more information. And then we can kind of use that to hone in more on, okay, we want to focus on certain cytokines, or okay, this looks like it might be a pathway we want to investigate more. So, every time we get a human sample, we try to get as much information out of it as we possibly can. We can use all the animal models and cell models that we want, but really ultimately to understand the disease, we need to be able to look at the human tissue and understand, what's really happening at that level, because a lot of times it's not the same as what's happening in the animals, because those eyes are so precious, and to us that's a huge gift that someone has given to science, and to our lab. So we try to get absolutely everything out of it that we can.
So the real challenge with this unique sample type is the volume, also sample prep.
Yeah, so with the human tissue, the biggest the biggest hurdle for us is getting the right postmortem time and getting things to us fast enough, but primarily the sample size.
Yeah, so you mentioned multiplex solutions as one of the things how you overcome these challenges. You mentioned the Procartoplex assays. You used the 65-plex assay. So thinking about biomarker identification, did you already have an idea what targets you want to investigate at the beginning of your research? Or how did you manage to identify your biomarkers of interest?
Right. So for us, it was really we want to see what information can we get? So that's why we kind of went with the Procartoplex, the 65-plex, to really get as much information. And I think that really provides a nice overview of most of the things that we want to see, and the advantage. We've also done some of the mix and match plexes where we've kind of created our own assays, which was nice because I can say, okay, we're interested in a few different things and kind of put them together that give us the proteins of interest, but does give us more of a targeted approach. So I think the ability to do the large scale and then narrow it down later on is just phenomenal. So still shocked that we can do this, look at so many. And I know now you guys have the 80-plex, but it's like, wow, being able to do that many out of a 25 microliter sample is just unreal.
So do you also plan to implement any orthogonal testings with other immunoassays for verification, for instance, ELISA? So any correlation data you want to
so we
obtain?
do plan on doing the ELISA, especially for the cytokines. I think we've done S100, and it was a pretty good correlation, at least with the patterns that we saw. And that's, the other way we would do it is looking, because we're traditionally an immunohistochemistry lab, we're interested in where things expressed. And if if there's a way to verify it with that, we will do that. The other thing is starting the quantogene eventually in the lab, and being able to do that parallel between the genes and proteins. And especially as we're working with some of these trickier tissues, that's where our human, I think for our human samples, that could be very, very valuable for us. So yeah, that's a technology that I really want to take advantage of in the future. The ability to do these multiplexes is spectacular. And I've seen the advances already in the last few years with your, different things being available with the Procartoplex and being able to mix and match a little better. So
yeah, that's really a huge advantage of this platform, I would say. So you can, on the one hand side, you can design your own panels, so you can address different aspects, starting with cytokines, implementing some angiogenic factors into that panel, some neurodegeneration markers as well. On the other hand side, you can use the same platform for looking into gene expression, as you mentioned. So, and we are already a little bit in the future. So looking ahead in general, what are you most enthusiastic about in your research field?
So I'm excited about where we're moving. We see more integration between academia and industry, but also between academic labs. And I feel like we're starting some exciting new collaborations that will really help us, understand our research better, but also, ultimately we do the science to help the patients. We have a conference where we actually attend with the patients. And that for me has been single-handedly the most inspirational thing as a scientist, because you see, you're interacting with the people that do the work, that they're the patients. They're the ones you're trying to cure their blindness. You see children that, know they're going to have a disease, know they're going to go blind in 10 to 20 years, and their parents. And with that disease, the parents are really, and the families are the ones driving the research. They're the ones funding it, and being able to interact with the parents for me has been the patients and the parents really a driving force to this is why we do what we do. And I think if we remember that, it really helps us do the science.
Yeah, I really agree. So the biggest inspiration and motivation is really have the feeling that one can make the world a little bit healthier. And this could already be kind of the final statement. I want to thank you so much for joining the podcast and spending some time with us, giving us insight about your research work. So thank you so much, Malia. It was really great to have you on this program.
Thank you. It's been a pleasure talking with you, and thank you for all the work you guys do.
Thanks for watching. I hope you can join us again at Be the Spark.